כאן. כן, בדיוק, זהו. אוקיי? ואני רק אחזיר לפעמים בעצם, כן.
Okay. Good afternoon, everybody. You can see I'm coming from Tel Aviv University, and this project included several partners. Our title is Quick Metabolic Engineering, and these are the main partners of the project. So, first of all, the project was conducted in Tel Aviv University in the Institute for Serial Crop Improvement, IIA. And our institute is focused on wheat research and using wheat wild relatives for wheat improvement for many, many, many years. As you can see from the title here, so in 2012, there was a G20 decision to support global wheat research because of the importance of wheat. And I don't think I need to tell you how important wheat is. So it speaks for itself. The main challenges, as they defined it, were climate-derived stresses, whether it is abiotic stress like heat and drought, or biotic pathogens, and they specifically mentioned a set of pathogens that were the rust pathogens that really great yield losses. That's what we are doing. The partner or the technology was from Professor Roshi Maeda, and he is doing world expert in metabolic engineering of plants, studying metabolomes and then using all kinds of technologies to improve plants or change plants and set of metabolites. And one of his inventions was that he discovered a new enzyme for the synthesis of tyrosine. Usually tyrosine is highly regulated, but he found an enzyme that is less regulated, namely the enzyme that produce, I'll show it in a minute, tyrosine is less regulated, so you can produce more tyrosine in the plant. As a result, you can have a lot of consequences that we'll talk about in a minute. In any event, this ADH alpha is the center of our technology here, and if you express this gene from wheat in different plants, like here, this is in tobacco, you can do it in soybean, other dicots, you can greatly increase the amount of tyrosine in the plant. And the last partner was Plant-Art Bio. It's a company from Israel that is looking for genes. They have their own invention for how to look, I would say, very, very efficiently for genes that can be used for whatever, mainly for microbes. They cover it from microbes, and they put emphasis on stress area. So just one word on the tyrosine, because that's the center of our talk today. So tyrosine is usually produced from a shikimate pathway, and this compound is then used, the arogenate is used to produce tyrosine. Two pathways, as you can see. We focus on this pathway. That's Hiroshi's invention. This is the enzyme, the true name, but we call it ADH. And usually the ADH is highly regulated by tyrosine itself. So the enzyme is regulated, not the gene transcription, but the enzyme is binding to tyrosine, and then it stops working. So the level of tyrosine is kept rather low in the cell. What he found, like I said, is an enzyme that is less regulated, so you can accumulate more tyrosine. And then in addition to protein synthesis, which is, of course, tyrosine is used for a range of useful metabolites. One of them is Betalains, and I highlighted it here because this is going to be our second main center of the talk today. So let's see what is the project is composed. First off, this is the objective. The objective was to increase tyrosine in wheat as a mean to increase, one, tyrosine itself, second, tyrosine-derived metabolites, and specifically betalain. We want wheat that produce all these compounds at high level. And I'll tell you why in a minute. The approach was to use genetic engineering to modify the wheat. First of all, wheat does not produce betalain, so we need to introduce the betalain cassette to wheat. Before this, we need to introduce the enzyme that is deregulated, so we can produce more tyrosine. More tyrosine will be used then to feed into the betalain cassette or fraction, and we'll get more betalains, that's the ruby cassette, more metabolites, and hopefully these plants will be also more stress resistant, because it's known that betalain confer resistance to abiotic and also to pathogens. So, technology, I already said, that's Hiroshi's invention, and we hope to engineer wheat with this DH5 alpha from beet, and hopefully this will result in higher level of tyrosine. Next, we want to add the betalain cassette into wheat, so we'll get red wheat. And then we want to combine these two and get even more betalain in wheat by feeding more tyrosine into it. Plus, from the plant art bio, we want to then take their genes. They have genes that they found, and as you can see, this is from their publications. This is control, and this is with the plant art bio gene under drought conditions, and you can see how much better the plant. So we want to combine because we will hope maybe there is a synergy between the betalain and their genes, so we'll get even a much better release. This is what we want. And we can do it with our technology because it's the ICCR. We have developed a Center for Wheat Genome Editing and Transformation, and we can manipulate wheat as much as we want. All this goes well. This is what we expect. So to get a super wheat with a lot of betalain and other metabolites that will have all kinds of benefits. It will have nutrition benefits. It will be resistant, hopefully resistant to stresses, both biotic and abiotic. So these are just a few results from our study. First off, we needed the first step to produce betalain in wheat. We got that, we introduced it into wheat. At the plants, we have very, very nice, even the quality wheat. It's really beautiful. And we have different colors. And because when you do a transformation, usually we use a tissue culture for transformation to get chimera, so we can even get plants that are half green, half red. So it's ornamental also. I didn't think about this direction in our project. Then the second step was to add the DH5 alpha. That's the enzyme, the deregulated ADH gene. Abigail, sitting here in the first row, she did all this fantastic work. She was a master student, so it's really much appreciated, so much to accomplish in one master project. And she generated different genotypes. I won't bother you with all the different genotypes, but these are for the control, so we can compare all these genotypes. And then after we got these genotypes, we characterize the metabolome, including tyrosine and betalain. And I just show here the tyrosine, and if you look carefully, you will see a very disappointing result. We don't see any major changes in the tyrosine amount in these plants. And we know the gene is expressed, no sure about that. We have all the controls in place, but no, there's no difference. We took 3 genotypes from each without it's clean experiment. So what's going on? As an approach, we had another backup approach. So this was one way. The other way we thought to change the tyrosine level was to take the wheat gene, modify it. Mutations that we know are the mutations that make the DH5 alpha less regulated, introduce into the wheat gene, and then put it back into wheat. And we did that. And surprisingly, in this case, I don't know if you can see it very clearly, but even if you just look at this this image here, which is a blowout of this one, we got tons of betalain in this tissue culture. This is in the callus. So it's so much that it's just moving back to the agar. Everything is right. completely red. Plus, here you can see, but the plantlets also are red. So also the plants, the mature plants are also much more red, meaning that probably we have much more betalain in this case. And Abigail generated these plants. She got the T0 plants. Now she got the T1 plants, and now we're waiting for the metabolomic analysis to see if there is more tyrosine and there is more betalain in this plant. Now you can ask yourself why. What happened? We also ask ourselves, our partner here he keeps working on this technology for a long time, and he keeps publishing. And unfortunately, he published this paper only 2 months ago. And if we saw this paper before, we knew, but now we know. Probably this is the explanation. We're not 100% sure, but we guess. He published a paper showing that the grasses ADH is even more deregulated than the beet ADH. So actually, we wanted to convert wheat to deregulated, but it was already deregulated. But the mutations we introduced based on the sequence of the beet and other plants into the wheat ADH made it even probably much less sensitive to inhibition by tyrosine. And probably this is the result, why we got so much more. So this is encouraging, but we're still waiting for the metabology. One more thing, I promised you that I told you that there are some indications that betalain confer resistance to pathogens and to stress. We tested our plants, and indeed they are more or less sensitive to these stresses. Here we see infection with rust, leaf rust. So you see the fielder line, which is the parental line. Then you see we use the Chimera TRF gene transformations. This is another control, anyways. So these are the controls. What you see is a germ, which are glutenine fused to green fluorescence. And this is the size of the colony inside the leaf. So you see the infection is more or less the same, but when you have the ruby plant, it's much reduced. And just to make sure that this is the case, we knocked out the ruby by CRISPR, and then you see the infection comes back. You can see it also in this panel. So the bottom line is that the ruby plants are much less sensitive to the pathogen, and similarly to drought. As you can see from the shape of the leaf, if you see the plant, it's really clear. When you put it in a soil stress, the red plants look much, much better. So all in all, there are good indications that it's a good phenotype. So these are our results. Now, what's next? First off, we need to finish some metabolomic analysis of those plants that I've mentioned. If we got them and we find that indeed the levels are higher, we will phenotype, of course, and find the consequences on the phenotype, different phenotypes to add. Plus, we are trying now to cross the best, we'll try to cross the best plants with the plantar biogenes, wheat, because we introduced plantar biogenes into wheat, again, by genetic engineering, and we'll see if there is any synergy between these traits. Here, but one minute. So I'll just tell you that if everything goes well, then we expect that we'll have higher tolerance to pathogen release, then we'll have more tyrosine, more antioxidant, vitamin A, all these metabolites derived from tyrosine. And also these red plants are very nice natural level crops. Because this doesn't work, it does. What are we going to, how do we see the prospect? I don't know. I'm not from the commercial side, but from talking to the company, we know they said if it's over 10% advantage, they will seriously consider keep supporting this development to the market. And with this, I would just want to thank the different people involved, the team from our institute, Abigail, first of all, sitting here, Anna, and several other people that I didn't put on the board. Then the University of Wisconsin, our partner, something, and of course, Dror from Plant Bio. I want to thank Shoshi from Ramot, from the commercial company from Tel Aviv, for her immense help. Noah sitting here from the Israel Innovative Authority, and Ofra Lotan also from the Innovative Authority, because Ofra was our examiner and she had very good remarks during the process that we incorporated into the project. And finally, of course, to the support from BARD, from Israel Innovation Authority. Thank you for listening.
Thank you very much, Amir. It's great work. Any question from the audience here?
A basic question. If the leaves are red instead of green, does that interfere with chlorophyll and energy? And why not use promoter to specifically make the ruby inside the grains?
Should I repeat the question? I think it's, of course, a very good question. If there are any side effects for the overproduction of betalain in the wheat, we didn't see it. We see normal development of the plants. In some cases, we saw a small effect on the root development, right? But generally, they are fertile. We didn't put it in the field, so we don't know. Chlorophyll-wise, we didn't measure chlorophyll, but I would expect there won't be any effect because there are other reports from other plants. When you produce betalain, it didn't affect the plant performance. However, putting it under a grain-specific promoter or anything like this is, of course, a very desirable thing to do, and we could consider it down the line, I guess, if there is any interest in this technology.
Any other question? Do we have question online? Okay, thank you very much. You didn't mention anything about you didn't mention anything about productivity. How much?
We didn't put it in the field, so we don't.
No, I mean in the greenhouse, the per plant, you see a reduction.
The spikes are
It's actually related to the previous question.
so the spikes are fertile, but you can the spikes they mature. They are grains. We can harvest them, and they don't show any difference from the fielder as far as grains. But I don't think this means much because you need to go out to the field to ask this question.
You're actually depending on chemical pathway, which depends on light, so it also might interfere.
Yeah.
Thank you very much, Amir. Again, for your don't run away, don't run, Amir. Please, I don't know if you meant if you have been here in the beginning. That's actually to symbolize the stage of your project. It's just sprouting, and we are looking for the blooming. The next time we really want to see the company with you here. And meanwhile, those are here, and that's for you to be in the field, to try in the field, doing work. Thank you very much. Okay.
You know that Yoram was my instructor in my team.
Oh,